Welcome back, students. So, today's topic of discussion is bacterial culture mediums, or you can say media. So, what are the different bacterial culture media which we use in microbiology laboratory? So, please do watch my video till the end and do subscribe my channel. What is culture media? Basically, culture media is a gel or liquid which contain a different type of nutrients which are beneficial for microorganism and which helps microorganism to grow. And they are also termed as growth media. Different microorganisms require different type of nutrients such as serum, blood, or there are some microorganisms which only require some simple nutrients like yeast extract, peptone, simple nutrients they require to grow. So, what is the significance of culture media? Culture media is used in order to identify the causative agent of the infected disease or the infectious disease. That's why culture media is required to isolate the microorganisms which are responsible for any particular type of disease or infection in human being, animals, or plants. Basically, there are three different category of media. First is media category based on physical state, that is liquid media, solid media, and semi-solid media. Second category is based on the presence of oxygen, presence or absence of the oxygen, such as anaerobic media or aerobic media. And third category is based on the nutritional factors such as simple media, synthetic media, complex media, and spatial medias. These are the three categories which have different type of media. So first of all, I would like to discuss with you media on the basis of physical characteristics, that is solid media, liquid media, and semi-solid media. Solid medias are those which contain about 1.5 to 2% of agar in it, and they are used in microbiological laboratory to isolate or separate the mixed culture of organism, or when we want to see the colony characteristic of any bacteria or any fungus. That time we use solid media. Liquid media are those which do not contain any agar in it. That's why they are known as liquid media, or also known as broth media. It is used for the profound growth of organism because they help in the fast growth of the organism, such as nutrient growth. Semi-solid media are those media which contain 0.8% of agar in it, and they are used in the microbiological laboratory to check the motility of the any motile organism or the non-motile organism, such as E. coli, whether the organism is motile or non-motile. That time we use semi-solid media. Then there is a simple media. Simple media are those which contain only single source of a nutrient such as peptone or meat extract. So 1% of meat extract or 1% of peptone makes up a simple media. Then we come to the complex media. Other media than the simple media are all media uh, excluding simple media are the complex media, because they contain some additive nutrients such as nutrient growth or nutrient agar, McConkey media, blood agar. They contain some additive nutrients such as yeast extract, NaCl, or we can say different dyes, etc., etc., serum, blood. Then that's why they known as complex media, because they contain a complex nutrients. Because some organism needs some complex nutrient, they or these type of organism are known as fastidious organism, because they have some special requirement for their growth. Then there is a synthetic media. Synthetic medium is a chemically defined medium which do not contain any yeast extract, plant extract, or animal extract, or they contain chemically derived nutrients such as Dobos culture medium which contain twins 80. These type of media are employed in investigation of such as metabolic needs, which have when we want to identify some metabolic product, that time we need some synthetic media. Then there is a anaerobic media or aerobic media on the basis of presence or of the absence of oxygen. Anaerobic media which require for the growth of anaerobic organisms such as Clostridium botulinum, Clostridium perfringens, etc. They, for example, Robertson cooked meat media, which contain a meat source, which which create a anaerobic environment for the anaerobic microorganism. Anaerobic microorganisms are those which do not require oxygen for their growth, and aerobic microorganisms require oxygen for their growth. Now come to the spatial type of media. First in this category is enriched media. Enriched media is made when we add some additive nutrient into the basic medium such as nutrient agar medium or peptone water medium. In this medium, when we add some additives such as blood, egg yolk, serum, this medium will become as a enriched medium. Enriched medium are those which require by some fastidious organism for their growth, such as Streptococcus. Streptococcus require blood for its proliferation. Then come to the selective media. Selective media are those which contain some of type of nutrient which will inhibit the growth of non-required microorganism, but uh, elevate the growth of required microorganism. For that example, bile salt agar. Bile salt act as a selective agent that will prevent the growth of other intestinal microorganism and favors the growth of Vibrio cholera only. Then come to the differential media. Differential media are those which help in the differentiation from the one bacterium to the another bacterium. For such example, Meconium agar. Meconium agar help in the differentiation of lactose fermenter and non-lactose fermenter organism. Lactose fermenter will look like a pink color colony, and non-lactose fermenter will look pale color colony. For that example, E. coli is a lactofermenter and Klebsiella is a non-lactose fermenter. Storage media. Storage media are those media which are required for the storing of bacteria for the longer period of time, such as excellent medium, chalk, cooked meat, broth. Then come to the enriched media. This media contains several type of ingredient that may either stimulate the bacteria being grown or suppress the other competitors, such as alkaline peptone water or tetrathionate broth, which are used for the growth of Vibrio cholera from the water sample. Then come to the transport media. Transport media are those which are required when we want to transfer our required microorganism from one laboratory to the another laboratory while preserving it for the longer period of time, such as storage transport media, which are required for the gonorrhea organism. Then come to the indicator media. Indicator media are those which will help in indicating the type of microorganism, such as McConkey media. This contain a neutral indicator which help in the identification of NLF and non-NLF organism. Another classic example is uh, sulfite containing Wilson blende medium, which help in the identification of Salmonella typhi by the formation of black color colonies or Salmonella typhi. Salmonella typhi will convert sulfite to sulfate. That will help in the formation of black color colonies of the Salmonella typhi. Then come to the sugar media. Sugar media contain 1% of sugar, which can be any fermentable substances such as glucose, mannitol, sucrose, and lactose. The generation of acid following the fermentation of sugar transforms the media into pink due to the presence of indicator. Also shows that gas is produced by a Durham tube is kept inverted inside the sugar tube and gas bubble are observed. These are the some commonly used media in the microbiological laboratory. First is nutrient broth, second is nutrient agar, third one is peptone water, and these are the composition of these mediums. Fourth one is blood agar. This is the most commonly used medium. It contain a defibrinated 5 to 10% defibrinated sheep or horse blood and add it to the melted agar medium having a temperature of 45 to 50 degrees Celsius. Blood act as a enrichment material or enrichment indicator or for the certain type of bacteria, and blood agar also help in the identification of hemolysis. There are three type of hemolysis which can be distinguished using the blood agar: beta hemolysis, alpha hemolysis, and gamma hemolysis. Beta hemolysis when there is a clear zone and complete hemolysis of the RBCs, which is caused by the Streptococcus pyogenesis. Alpha hemolysis is when there is a greenish zone or around the bacterial colony. Uh, and gamma hemolysis is when there is a no hemolysis caused by the organism. There is a chocolate agar medium which is required by the pneumococcus, gonococcus, meningococcus, or Haemophilus influenza as well. Then there is a McConkey agar, which is also a selective media, indicator media, and differential media, as I told you in previous slides. Then there is a Mueller-Hinton agar, which is used for the antimicrobial susceptibility testing. Then there is a his serum water medium, which is used for the growth of pneumococcus, Neisseria, and Corynebacterium diphtheriae. Then there is a Lowenstein-Jensen medium, which is used for the Mycobacterium tuberculosis. Dubose medium is a synthetic media which is also used by the Mycobacterium tuberculosis or other Mycobacterium species. This media contain a 280 bovine serum albumin, casein hydrolysate, asparagine, and other salts. Then there is a Loeffler serum, which is used for the growth of diphtheria bacilli, and it contain a serum of horse, sheep, or ox or rabbit. And then there is a telluride blood agar medium. It is used for the growth of Coryn bacterium diphtheria. Please do subscribe my channel and like my video, and thank you for listening me.